Okay, so welcome back to the fourth part to genetics section 1.2 in your notes. So to start off today's lesson, we're just going to quickly go through the answers from page 34 and 35 in your notes. Feel free to pause if you need a little bit of extra time to process, or skip through and check your answers if you feel like you're noticing that they are correct. So the focus of this practice is on complete dominance. So this is where one trait is present and it is dominant over the other one. So you would need both alleles present in order to show the recessive trait. So let's do this kind of together here. So identify the phenotypic ratio and the genotypic ratio of the offspring. So we have two parents. One is dominant, TT, capital T, sorry, and the other is also showing the dominant trait, but it's capital T, little t. One T from here, and the second T from here. One T from that first one. One T from there. So both of those are capital T, just like the parent. Capital T from the top, lowercase t from the side. Capital T from that second top one, lowercase t from the side. So what we're seeing here is two homozygous dominant for tall and two heterozygous offspring. Now, phenotypically, this is 100% tall. And then the genotype, though, is 50% homozygous, capital T, so the dominant, to 50% heterozygous, so capital T, little t. So again, 100% of these are showing the tall phenotype because the dominant capital T is present in all of these. Okay, I'm just going to quickly fill in the second example and then we'll go through it together. Okay, so I have gone ahead and filled in the second example here for you to look at, and I've also color-coded it to help you to differentiate which parent gave which allele. So looking at this, we can see that we have two heterozygous, and sometimes it's easier to start with the genotype over the phenotype. So with these questions, you don't necessarily have to do it in the order shown here. So we've got 50% capital T, little t, so the heterozygous, to little t, sorry, to 50% of the homozygous recessive, little t, okay? And you can see that it's a 50-50 split with the offspring. In this case, because only two individuals out of four have the tall allele, only 50% are tall to 50% short. So again, just a reminder, when you are filling in the Punnett square, make sure you always put the dominant allele first if it is present. So this is just a convention that is required in order to do the Punnett square correctly. Okay, so here are example 3 and 4 from your notes. I've again tried to color code these to make it a little bit easier to visualize. So we have a tall heterozygous parent crossed with a second tall heterozygous parent. So the result of this is you can see that 75% are tall phenotypically and 25% are short. So 1 in 4 are short, as that is the recessive trait. So this is based on the genotypes. If we look, we can see that only one of the 4 individuals, so 25%, is homozygous dominant, TT, both capitals. 50% of them are heterozygous, big T, little t, and 25% are short, which is the recessive trait, little t, little t. For the next example, if we cross two short parents, together, because this is the recessive trait and there's only little t's present, we're going to have 100% short. 100% of the genotype will be homozygous recessive, little t, little t. So is it possible for two tall plants to produce only tall offspring plants? What genotypes would the parents need to be? So yes, this is possible, and there's two different possibilities to have all tall offspring. So we can have homozygous capital T, and homozygous capital T crossed. So that would give you 100% capital T. So all tall, or homozygous dominant capital T crossed with the heterozygous capital T, little t. Number 2, is it possible for two tall plants to produce short offspring plants? What genotypes would the parents need to be? So again, yes, the parents would need to be capital T, little t, so the heterozygous, and capital T, little t. So as long as they're both heterozygous. Is it possible for two tall plants to produce only short offspring plants? Why or why not? So technically, yes, but statistically very unlikely. So you could have extreme luck and get only 25%, which would be the recessive short from this particular cross, but is statistically much more likely to have tall offspring because there is a 75% chance of that happening. So getting the 25% is much more likely to happen in an animal over a plant, so that's important to know. Mainly because mammals, for example, don't usually have hundreds and hundreds of babies. So it's statistically closer to that 25% short and 75% tall split in plants. Okay, last two questions before we move on to our notes. So is it possible for two short plants to produce tall offspring plants? Why or why not? So no. Since there is no dominant alleles, there is a 0 chance that they could produce a tall offspring, as the tall allele is the dominant allele. Question 5, why do we refer to the tall, capital T, allele as being dominant and the short allele, little t, as being recessive? So if big T is present, it is expressed, and if little t is present, it will only show if both alleles are the recessive little t allele. So the dominant allele dominates over the other one, so the short allele is only present if both alleles are short. So remember again that eye color is actually quite complicated, but for the sake of this course, we used eye color to show complete dominance. For fur, remember that the black fur allele was present. When the black fur allele was present, that would mean that the fur is black, and that's what would be shown. White fur was only present if both alleles were for white fur, as it is the recessive trait. So other types of inheritance. Mouse fur color and eye color are examples of complete dominance. There are several other types of gene expression. These are incomplete dominance, codominance, and sex-linked dominance. So we are going to talk through each of these one at a time, starting off with codominance. So in codominance, both alleles are expressed, meaning they both show up visually. What does that look like? So one capital letter is given to the gene. So for example, F is for feathers. Here we're going to be working with a chicken as an example, with different superscript letters, meaning they sit up in the exponent position for each allele. So for example, B for black, W for white. So what this means is that you could have your feather gene, FB would mean black feathers. FW would be white feathers. And if you had FB and FW together, we would have what is called checkered feathers. So 2 copies of the black allele means that you would have black feathers. 2 copies of the white allele means you would have white feathers. And if you have 1 copy of each allele, so F, B, F, W, you end up with what is called checkered pattern. So this shows as spotted, speckled, depending on the species, but essentially you're seeing both colors present. So this is not the case for all chickens, but it is for this particular type. An example here, in certain chickens, black feathers are codominant with white feathers. Heterozygous chickens have black and white speckled feathers. What would be the offspring of a homozygous black chicken? So FB mates with a homozygous chicken, FW. So we've written out our cross. So FB crossed with FW. We draw our Punnett square. This parent donates the FB. This parent donates the FW. FB from here, FW from this parent, and so on. So we end up with 100% FB, FW, meaning the checkered phenotype would be 100% of the chicken offspring. So all of them are showing the FB, FW phenotype, meaning that they are all visually checkered, or whatever you want to call that checkered pattern. So both feather colors are seen. Okay, next example, what would be the offspring if a checkered chicken, FB-FW, mates with a homozygous white chicken, FW-FW? So to start, we write out our cross utilizing these symbols. So FB-FW with FW-FW homozygous. Then we are going to draw out our Punnett square. So FB, FW is one parent, and FW, FW is our second parent. So we get the FB allele from this parent and the FW from the second, FB from the first parent, FW from the second. Both parents here contribute FW. So we've got a homozygous and another homozygous. So looking at our genotypes then, 50% are FBFW. And 50% are FW homozygous. So they would be white. So then from here, we can then determine our phenotype, which is the visual appearance. So 50% are checkered. And 50% are white. So essentially, you are getting a 50% chance of looking like one of the 2 parents. So some other examples of codominance. You can have a chestnut horse crossed with a white horse, and then you'll end up with a roan colored horse, which is basically just a lighter brown with a really light mane. Flowers, you may see multicolored flowers, so like the example of the pink flower being crossed with the white, the pink and white kind of speckled flower results. And additionally, in the notes, you have another example on red blood cells. So you can have sickle cell anemia. You can have an allele for regular cells and sickle cells. And if you have both alleles, so one of each, you'll have both blood cell types present in your body. If you had normal alleles, you would only see the normal blood cells. Or if you had 2 copies of the sickle cell allele, you'd have sickled celled red blood cells, which can make people really sick. But if you only have 1 copy of the sickle cell, you'll be fine. So this is the end of this lesson. I would like you to now move on to the practice, which is on page 38 and 39. We will go through these answers in the next lesson.